Hello, and welcome to part 2 of our short bit of history of microbiology. All right. The next 4 scientists I'm going to kind of go over briefly are going to be Joseph Lister, Fanny Hesse, Robert Koch, and Esther Letterberg. Okay. So we're kind of going around the same time now with Joseph Lister as when Louis Pasteur was working. Joseph Lister was credited with the concept of being aseptic during surgery, all right? And science-wise, anytime you see something that starts with an A, it typically means without. In this case, septic relates to contamination. Because believe it or not, prior to 1867, physicians didn't wash their hands. They certainly didn't take precautions before bringing a person into surgery. But what Joseph Lister discovered is that if you prevent or reduce the amount of bacteria in the environment, patients survive surgery, and that tended to be a good thing. Okay, so what Joseph Lister did is he had a mist of a phenol compound that was released in the surgical suite, and that actually reduced the bacteria because the compound was toxic to bacteria, all right? And as an extension of that, part of what goes along with it would be washing tools between patients so that you don't move bacteria between individuals, okay? So the concept of being aseptic as a physician was kind of brought home by Joseph Lister. That was where that first came into play. And now that's part of our routine medical care, because now certainly you wouldn't have your surgeon going straight from the morgue right into the surgical suite without washing their hands first, okay? So part of controlling microbes in our environment are contributed to Joseph Lister, all right? The next 2 scientists I'm going to talk about are Fanny Hesse and Robert Koch, okay? Now in textbooks, you tend to see this name a lot because what Koch is responsible for are something known as Koch's postulates. So what that is a series of steps that scientists go through to determine if an individual bacteria is the cause of disease. Because Robert Koch showed that tuberculosis, what we call TB, was caused by an individual bacteria. And that individual bacteria is Mycobacterium tuberculosis, okay? But Robert Koch would have never been able to show that association if it weren't for Fanny Hesse. Because Fanny Hesse was involved in the process of creating the media that we now use in modern microbiology labs. And Fanny Hesse wasn't necessarily a trained scientist. Her husband, Walter, was. And she worked with her husband. But with Fanny's experience cooking, she discovered that the substance agar worked as a much better culture medium than what they were using at the time, and what they were using at the time was gelatin. Gelatin for the bacteria could dissolve that gelatin and kind of liquefy it. Agar didn't have that property about it. So in 1881, Fanny Hesse introduced with her husband in their lab the use of agar and agar plates to culture what they couldn't see, to culture those microbes. Robert Koch used their materials, although never actually credited Fanny Hesse, which is why I have Fanny in here, because she deserves the credit for a lot of what we do in modern microbiology, just by simply coming up with materials that we can use. So she discovered the material that allowed Robert Koch and subsequent microbiologists to isolate individual bacteria. And if you take the microbiology lab, you're going to be using media that has agar in it, okay? And that goes back all the way to 1881, all right? As a side note with this, I put the dates up here for your information. On the tests, I'm not going to ask you the dates. What I want you to take away from the history part is how we had a progression from not being able to see the bacteria to being able to see them and culture them and associate individual bacteria with disease. So if you follow along the names with what they're associated with, that's what I want you to take away out of this particular set of videos, okay? So we've got Lister, Hesse, and Koch, all right? The final scientist I'm going to talk a little bit about, we're going to kind of jump ahead here to the 1950s. Because Esther Letterberg was involved in the early field of bacterial genetics. And in this course, we will cover bacterial genetics in a later learning module. But a lot of the information that we're going to go over in those later modules came from work from Esther Letterberg and her husband, Joshua, okay? But what Esther was responsible for as part of her PhD work is she discovered viruses, well, she didn't discover virus, she discovered a virus, and that virus is called the Lambda virus, that can specifically infect bacteria. And those viruses had the ability to transfer viral DNA. Oh, this is a squeaky marker, I apologize. To the bacteria. And today we know that process. Oh, let's switch colors. Maybe we'll find one that's less squeaky. As the process of transduction. And we'll discuss transduction in a later video, okay? But her work with the Lambda virus, and also later, we'll go over this again when we get to this. She also was responsible for discovering what's known as the F factor, or the fertility factor in bacteria. And what the F factor does is it allows the formation of pili in bacteria. And those pili, again, are used for the transfer of genetic material. So Esther Letterberg was one of the early, very early bacterial microbiologists who also studied genetics, so the transfer of DNA, okay? Hopefully that kind of gives you a feeling for how the process or the field of microbiology has kind of progressed. We're going to cover in subsequent learning modules a lot of these processes that we've talked about. We'll talk about pasteurization. We'll talk about how bacteria are associated with disease processes. We'll talk about bacterial genetics. All right, so if you have any questions about this, please let me know. Thank you for your time.